So we should share genetic information. And just to illustrate why we need to share genetic information, I would like you to meet this adorable young kid that was born with golden hair syndrome and developed also medulloblastoma during her childhood. The family of this girl contacted my group for help. They heard that we do exome sequencing and wanted to see if we can help her. We sequenced her entire genome and were able to identify the causal mutation for the golden hair syndrome. And quite likely this mutation, its OTX2 duplication, also contributed to her predisposition for brain cancer. Now she's okay. She celebrated 2 weeks ago her bat mitzvah. She's in remission. And the only reason, of course, that we could identify this duplication was because we could compare her genome with the genomes of thousands of individuals from the 1000 Genome Project and other projects that are healthy, and we could contrast this genome to identify the causal mutation. So this is kind of like where we start, and we need to find ways how we can share these genomes while protecting the preferences of our participants. My interest in privacy goes to the days that I was an undergraduate student, and I worked as a hacker in a computer security company. We used to be hired by credit card firms and banks to conduct penetration tests, to try to hack into their systems in order to identify critical gaps and work with them to improve the security measures that they have in their bank. And what I'm going to show you here is one of my favorite hacks. What you see here, this is the younger version of me. And behind me, you see the door to the IT department of a major bank in Israel. Now, this door is controlled by an intercom, a very simple device. You press on the button, it calls the secretary, and if she knows you, she would press 8 on her phone to open the door. The amazing thing is that each one of you can open this door in 5 seconds by playing the sound 8 from your cell phone just next to this intercom. So currently, it is 10 p.m., there is no secretary, and let's see how I can break into the building. And hopefully the audio is connected to the presentation. Okay, calling the secretary. Playing 8 and taking the money. Don't try this at home. So after we do this penetration test, we can go to the security manager of the bank and have a constructive discussion how we should move forward and to find a solution. And this presentation is modeled after this penetration test. I'm going to show you in various cases how we can break into genomic data, and later on, I would like to discuss with you what you think, how we should move forward with the current gaps that we have. So kind of like the presentation, most of what I'm going to present, we published about 2 years ago in Nature Reviews Genetics. It's a systematic review of methods to breach genetic privacy. We also wanted to discuss methods to protect genetic privacy, but we realized during writing this manuscript that there are not so many options to protect genetic privacy. So if you're interested to learn more about what I'm going to present, everything is in this manuscript. So in general, when we talk about threats or attacks to genetic privacy, we talk about 2 different routes. In the first one, we have identity tracing. We have a genome that is allegedly anonymous, such as the 1000 Genomes Project. There are whole genome sequencing, we just don't know who's this genome, and we want to find the identity of this genome. And another option is what's called attribute disclosure. In attribute disclosure, we have already DNA of someone that we know, and then we will take this genome and we'll try to search using this genome in some GWAS studies to infer phenotypes, a hidden phenotype of a person. Today, I'm just going to talk about identity tracing attacks. So we're just going to focus on this part at all, but in the review, there is a section about all these attacks over here. Okay, so identity tracing. What is the concept of identity tracing? First, let's think about what's the concept of anonymity. Anonymity means that you are basically lost in the crowd. If there are many individuals around you and someone cannot distinguish between you and the other individuals, then it means that you are anonymous. So the whole point of identity tracing attacks is to remove the crowd from the person, to start to basically, if we have here the crowd, we try to separate the crowd into smaller groups and smaller by some identifiers until we get to a specific individual. So that can be, for instance, we go here and we say, okay, we know that our person, the genome that we are looking for, came from a person in European origin. Okay, so that means that I can separate all the people on Earth and just inspect the people with European origin. And then let's say this person was collected from the Netherlands. So now I can separate this European origin just to Netherlands, and I can keep going until I get to my person. So this is the concept of identity tracing. Now the question is, when I do this separation, we want to quantify what are good identifiers and what are bad identifiers. And for that, we're going to use entropy. You don't need to focus on the quantitative aspects. The point is very simple. Entropy means that we're going to measure the number of possibilities in bits. So basically, if we take the population on Earth, 7 billion people, then if we just measure it in bits, how many bits of information we need to gather to identify a person, it's only 33 bits of information, which is quite small, and I'll show you why. Let's look at different types of identifiers that we can learn from genomic datasets. For instance, sex. You can know if a genome has a Y chromosome or not, right? And that means that we can half the population into two parts. Sex will give you 1 bit of information. So if we need to identify every person on Earth with 33 bits, if we know the sex, we just need 32 bits. Now, if we know, for instance, the eye color of the person, and this refers to the US population over here, it means that we have about 1.4 bits of information. If we know the height of the person, let's say that we know the height in the resolution of 1 centimeter in the population, by knowing the height of the person, I gained 5 bits of information. So by adding more and more identifiers, let's say someone that is a male, has blue eyes, and is 180 centimeters, that means already that I narrowed down the search space and getting closer to this person. Okay, now today I'm going to show you several attacks, but I want to focus on how we can infer the surnames of individuals from their genome. Usually when we put genetic information out there, such as in the 1,000 genomes, do we put the surname of the person? No, we don't put. But I will show you how we can derive the surname of someone by inspecting the genetic information of this person. And this is the first attack that we're going to talk today. Okay, so we know for over a decade that there is a correlation between surnames and the Y chromosome. For instance, here we have the Smith couple. And if this couple is having a kid, the father will give his kid his Y chromosome, and in most Western societies also his surname, Smith. Now, if this son is getting married and also has a son, he will give him his Y chromosome and also his surname. And now you see it starts to create a correlation between the Y chromosome haplotype and the surname. Genetic genealogy companies are aware of this correlation, and they offer services. They will send you a swab to sample the DNA in your cheek. You'll put it in an envelope with $100, very important. Send it to them, and then they will genotype a series of YSTRs, short tandem repeats on the Y chromosome, that look like that. Short tandem repeats, just basically there is a small motif that is repetitive, and they will measure this, the size of this repeat, and will put this data with the surname on the internet. There is a database called ysearch.org. This database is available to each one of you right now from this room without subscription. It's free. And what you see over here are my own test results. Why do people do that? Why do they are getting tested? Because it's a lot of fun. You can learn about your ancestry. You can find your patrilineal relatives and even meet the black sheep in your family. So many people conduct these tests, and we have these very nice databases. The question that my team asked was, can we now take whole genome sequencing data from someone that is allegedly anonymous, search these databases, infer the surname of the person, and triangulate this individual? So our algorithm has basically 3 steps. First, we have a pre-processing step. We need to extract short tandem repeats from the whole genome sequencing data. After we have the haplotype of these short tandem repeats on the Y chromosome, we will search the genealogical database We'll infer the surname, and then by combining more information, we'll triangulate a single individual and get to his identity. Let's see how it works. Okay, so to get short random repeats, we need to, they are a bit, not very easy to genotype with regular tools for high throughput sequencing. So we developed a few years ago Lobster. This is a tool to to genotype short random repeats from high throughput sequencing. We published it in Genome Research a few years ago. We have also a website that you can download this tool. If you like us, if you find this tool useful, please like us on our Facebook page. And just to show you that it works, here is the repeat number that Lobster reported, and this is the difference from the human reference. So negative means -10, here means -10 nucleotides from the human reference. And here is gold standard capillary electrophoresis for these short random repeats. The data was taken from the Human Diversity Project that conducted this capillary electrophoresis genotyping of STRs. And this is what Lobster reported for the high throughput sequencing data. And there is a very high correlation between these two methods of 95%. So it works pretty well to get short random repeats, especially from the Y chromosome that is hemizygous. We don't need to deal with the diploid nature. So we get very clean genotypes. Okay, so we have now these STRs. Now we need to search the databases and find the person with the surname. So we decided in this study to focus only on two databases that are out there, ysearch.org that I mentioned before and smgf.org. Together, these databases at the time of the study had 140,000 records of surnames associated with a Y chromosome. Okay, so how are we going to find the surname in these databases? So kind of like, let me give you an intuition. Here is our target. Let's say a genome from the 1000 Genomes project. And now we're going to scan each record in these two databases. And we're going to estimate, using the STR markers, the estimated time to the most recent common ancestor between these two genomes. Now, if the time to the most common recent ancestor goes to thousands of years ago, let's say to the ancient Greek or something like that, we will say probably these two people don't share the same surname because the surname system was invented only 500 years ago. So there is no chance that they actually have the same surname. But sometimes we are going to be quite lucky and we're going to find a close relative, maybe 8 generations ago, that they have the TMRCA between these two individuals. And then if they are close relatives, we will say that the surname is much more likely that they will share between them. And we're just going to scan the entire database to find the most likely surname. In a more quantitative way, basically what we use here, this is from Walsh. We're going to estimate the time to the most recent common ancestor using this Bayesian approach over here that takes into account the effective population size and the mutation rate of each STR. Then we're going to find the surname with the shortest TMRCA. Then we're going to calculate some retrieval score. And basically this means kind of like, let's take the best surname that we can, the best record that we have, and the second best record, and see if there is a strong difference between them in terms of TMRCA. If there is, we have more confidence about the accuracy of the surname prediction. And finally, if this confidence score that we have is above a certain threshold, we will report a surname, or otherwise the algorithm will say, I don't know. And this threshold, the user can specify any confidence if you want to be very aggressive or be very precise about the surname inference. But the take-home message is that the algorithm will have basically two types of answers. Either it will give you a surname, or it will tell you, I don't know, I didn't find anything. Okay, now we tested empirically to see if we can find surnames of people. We took a person that, people that we knew their surname, and we also had this YSTR haplotype. We queried these two databases with this method, find the most likely surname, and compared the surname that we identified to the surname of the person. And we repeated this process to gain statistical power about 900 times. And we also used some stratified sampling approach just to make sure that we represent the US population in this query, that our results are unbiased. What did we find? We found that for a US Caucasian male, the probability of retrieving a surname is 12%. For 12% of the US population, I can do this process and get the right surname. Then 5% of the cases, I will get a wrong surname, and 83% of the time, the algorithm will tell me, I don't know. I don't have enough confidence in what I identified in this database. Now, you might say that 12% is quite low. But think about the precision medicine initiative now that we have in the US that aims to sequence 1 million genomes. 1 million genomes, it means that we can do surname inference for over 100,000 genomes. And that might be a problem. Okay, so now we have the surname, and the question is how we can triangulate the person. So I want to emphasize that you might say, okay, if you have the surname, but probably just all these like common surnames in the US, Smith, Johnson, Jackson, there are millions of people in the US with the same surname. So it doesn't help you so much. But in fact, what we found was that in most cases, the surname that we recover is quite rare. It is found in 40,000 males on average, each surname. So think about the search space in the US is 150 million males when we start the process. By having the surname, I reduce the search space to about 40,000 males. So this is a quite strong reduction. Okay, so that's nice, but it's still 40,000 males. How can we get to a specific person? So we thought about the following process. In the US, the HIPAA, which basically this is a privacy rule that regulates health information, allows to publish the age and the state of the person. And you have it in many genomic databases. You see where the sample was collected and what was the age of the person. So we thought, what would happen if we have the surname, the age, and the state of the person? How many people would match such a profile in the US? So we simulated basically different ages based on the US census, the density of the population of each state, and the surname that we recovered. So let's say we took someone that is 40 years old, lives in Colorado, surname's Adams. How many people would match such a profile? We did a simulation, repeated it 100,000 rounds until it converged, and what we found was that if I know the age, state, and surname, I can get to 12 individuals. This is in most cases. We get just to 12 males. When we have 12 males, from now on, every other piece of information would identify the person. Think about it. We can even just call these 12 males and just ask them, did you participate in a genomic study? And by social engineering, they might tell us if they participated. It becomes tractable at this point. Okay, so we have this algorithm, but I just showed you kind of like in patches that it works, right? I showed you that we can recover YSTRs. I showed you that we can recover surnames. And then if we have the surname by the simulation, I showed that we can get very close to the person. Let me now put everything together. So to do a test case, we downloaded the genome of Craig Venter. And this genome is available from NCBI. Then we ran Lobster on his genome and recovered all the YSTR markers that we could identify using Lobster. So for instance, here are the raw results for DYS 458, and we identified 17 repeats. We went to ysearch.org, and then for each marker that we found, we just put the number of repeats. And then we clicked search on Y-chr, and after a few seconds, we found that Venter was the top match in Y-chr. So I just showed you how we can take whole genome sequencing data and identify the surname of an individual. Now, remember, everything here relies on public resources. So if you want to try it yourself, you can go to this link, bit.ly/find_Craig. This link will redirect you to Y-chr with all the markers that we identified on Craig Venter genome. Click search and see that you can find Venter as a top match. There are no secret keys over here. Everyone with an internet connection can replicate these results. Okay, so we got a surname from whole genome sequencing data. But you might ask, okay, so you got Venter from Craig Venter genome, but there are probably thousands of Venter in the US. Can you get to Craig Venter? And the answer is yes. We know that we recovered the surname Venter. We know that the state of residency is California. This person was born in 1946 and is a male. We can go to a website called ussearch.com, and this is a public record search engine. And you can put these identifiers, and if you put them, what you get is there are two records that would match this profile, one of which is our friend, J. Craig Venter. We see where he lives, all his former wives over here. And, and you can also, by paying $5, you can get a more detailed report with his email account and telephone, but we didn't have budget for that. We just used the free version to very low budget. It's amazing what you can find online, right? That there are these like databases that you can put age, state, and the surname, and you basically just see records of people. I'm also one of these databases online. So quite interesting. Okay, so now we got Venter, and you can say, okay, that's exciting, Yaniv, but come on. When you started the process, you knew that this genome belongs to Frank Venter. Can you show me that you can identify genomes of people that you don't know beforehand? And the answer is also yes. We took genomes from the 1,000 Genomes project. We focus on the CU population that represents the US, and these are from Utah. We took 10 genomes. We used Lobster to recover the short random repeats, queried these two databases, and then we got surname predictions. For 8 out of these 10 genomes, the surname predictions matched people from Utah, which is pretty good. But did we find the right family? So first, let's focus on this pedigree over here. This is a 3-generation pedigree. We recover the surname of the paternal grandfather and the surname of the maternal grandfather over here. We don't give the full details of this pedigree just to respect the privacy of this family. After we recovered the surnames, we went to Google, and we did a very similar query to what I show you over here. Again, not the exact details. And once we queried, we found an obituary that matched exactly the description of this family in the 1,000 Genomes and project and the Criel database that stores their cell line information. What do I mean by that? First, the surname of the father in the obituary matched the surname of the paternal grandfather. The maiden name of the mother matched the surname of the maternal grandfather. The number of kids was exactly the same, and remember, this is a large family. The birth order of males and females was exactly the same. Think about it. It's like flipping a coin multiple times and be able to predict the flips of the coin. This family is also the obituaries for a family in Utah. And we also checked the ages of some of the individuals in the obituary and versus what we know about their ages in the 1,000 Genomes project. It was exactly the same. So we calculate the probability that we got the wrong family after scanning all households in the United States. And the probability, the upper bound, is less than 5 times 10 to the minus 9. Very, very small probability that we got the wrong family. At this point, we knew their entire identity and all their genomic data sets. We also, for some people that are younger, we also had their Facebook account with photos and everything. Okay, so, but then you might ask that this is beginner's luck. So we identified one family. And in fact, when you submitted the paper, the reviewer said, okay, that's beginner luck, show us that it works. So the paper was rejected initially. So okay, we have to do it again. And we did it again. And we identified another family from the 1,000 Genomes and another family from the 1,000 Genomes. Each time, the probability of a random match was extremely small. In fact, we had so much information at this point that we could see what is the relationship between the people that donated the Y chromosome to this fun genealogy project and the people in the 1,000 Genomes. And I want to emphasize a critical point. These are not the same people. We don't need the same person in both databases. For instance, here, this person was in the 1,000 Genomes. His second cousin, once removed, was in the genealogical database. Just wanted to know about his ancestry. But the information travels through these deep genealogical ties to identify this family over here. How many of you know your second cousin once removed? And know if they donated their data to genealogical databases? Even if you don't put your data out there in these databases, your family members or some distant relatives can do that, and by that identify or reducing your genetic privacy. So in total, we were able to breach the privacy of about 50 CU samples in the 1,000 Genomes project. So we published this paper a few years ago in Science, and before we published the paper, we contacted the NIH and told them, listen, we found this gap. We don't want to go first to the press with this or to publish the paper before we let you know to see how we can fix this gap or at least get your attention to it. And so they asked us just to delay our publication so they can write a perspective about it, that it was published back-to-back with our paper. And Nature editorial thought that this is a very nice way to address security loopholes in genomics. And this, I have to say that this is very standard in hacking, in white hat hacking in general, in the computer security world, that first, before you go and publish your hack, you go to the company that manufactures the device and tells them about it and let them give them some time to react to this gap. And also, we're pleased to see that this study generated a lot of media attention because we wanted to initiate these discussions between the general public and between scientists about how we should move forward with genetic privacy. Okay, so we talked about surname inference. So this is one type of attack. We can look at the genomes of individuals and get there and identify them. Okay, now let's talk about another attack, which is called side channel leakage. In a side channel leakage attack, what happens over here is not that we basically put identifiers as part of our data, and we just don't know that we put these identifiers per se. We forgot about them. Let me give you an example before we go to genomics. Okay, here is a photo I took last night. I just downloaded a random photo from Flickr. We just search for a photo and download this photo. Now, this person looks, how many of you think this is an anonymous person in general? Raise your hand. Okay, the face is covered. Come on, we don't see anything over here. And this is not just a regular JPEG file. What you don't know, or some of you might not know, that in a JPEG file, there is an embedded information called EXIF that describes some metadata, where the photo was taken, by what camera it was taken, and so on. So if you look at the EXIF data, you see here that actually we have the location of the photo, and we also have the serial ID of the camera. Now, if we take this location and we just search in Google, so this was in some lodge in Maine, if I remember correctly. This is the reason why I put the full location, because it's just a public place. But now we know where the person was when he took this photo. But the story continues. So, right, we can take this, we have the serial ID of the camera, right? So there is a database called Stolen Camera that you can put and you can search for serial numbers of cameras. Now, this database is super cool. It just goes online, crawls different photos, like photos online in Google Images, and tries to find serial numbers. So if your camera was stolen and someone uploaded a photo online, you can say, aha, someone already took this photo, and see who is this person and try to find your stolen camera. So this was pure luck. I put the serial number that I found from this camera in this stolen camera finder, and surprise, surprise, the camera was stolen a few months after this person took the photo in this lodge in Maine. So kind of like the emphasis of this very small exercise is that when we have rich data and we don't know what exactly in this data, there might be some identifiers that just cool on to this data. And this might have some consequences for privacy. Okay, so what's the relationship to genomics? There is the Personal Genomes project by George Church in Harvard. In this project, people are getting whole genome sequencing. They put information online, but they also allow for people to upload their 23andMe results. So if they have, kind of like people have their own profile that is totally open, they don't put their name, but they can put more information about themselves, such as phenotypes, but also contribute their 23andMe data if they want. Yeah. Thank you. Okay, so this study was done by Sweeney et al. In 2013. And the problem is that when you download your 23andMe data, so here is my own 23andMe download page, the name of the file contains your first and last name. So I just took it yesterday, or a few months ago. So here is my genome over here. And you see this is the default file name. So what happens with default? People don't try to change their default, right? If this is the file name, what happened is that you can go now to Personal Genomes, you upload your data, now your file name has your name. So if you go to Personal Genomes, this again, I just took yesterday one data set over there. So you just download the data set from the Personal Genomes project. When you download the data set, everything looks fine, right? There is some weird number over here that doesn't tell you anything about the person. But if you unzip this file, surprise, surprise, we have the first name, and I didn't put the last name just to respect the person's privacy. But we have the first name and the surname of the person. So here is a database that allegedly tell people don't put your first and last name. but allowed people to upload their 23andMe data. They never took any measures to change the file names. In fact, they just put the file names inside they just zip them into another file, but this looked that everything is fine. But if you unzip, then you find this person first and last name. So it's even worse. It gives you maybe the first impression that everything is fine, but it's not. And this is what's called side channel attack. So if sometimes you take some information from your participants, you put this information without any step to sanitize your data, it might embed hidden identifiers of your participants. Think about a Word document. A Word document has the properties field into it that most of you are unaware of or maybe don't know but don't think about it. In this properties field, it says who is the author. So if your participant maybe wrote something for you and just put this data without copy and pasting to a new file, it has his identity already in the file. Okay, so side channel is another attack. Now, let's go back to this list of identifiers. There are many identifiers over here, like surname, that maybe are not explicit in the data, but we can find them by genetic inference. Let's think about height for a moment, right? So maybe we don't publish the exact height of our participant or our patient, but if someone has access to the genetic data, they can infer the height and get the height of the person, right? So that might be, and this is called phenotypic prediction. But let's see if it works. What I show you over here is the information content of height. Just look at the black line over here. This is the information content of height versus the accuracy of the prediction that we have. Now, if we have perfect knowledge, if we put the exact height of the person, really the number we measured, and we put the number with the genome of the person, we get about 5 bits of information. But, of course, we cannot predict perfectly height because the upper bound on prediction is the heritability. And the heritability of height is 80%. So 80% means that now, instead of getting 5 bits of information, we got about a bit more than 1 bit of information. But wait, this is just the heritability of height. What we know about heritability? It's missing. We don't know the heritability of height. We know right now with the best genome-wide association study published in Nature Genetics 2 years ago, we know something about 16% of the variability of variance of height in the population. So what we get from height prediction, we get not a lot. We get something that is close to nada, zero, nothing, garnished. So just to show you kind of like, it's about 0.1 bits. This is what we get from height. So the sex of an individual tells us 10 times more than what we can do from height prediction. So height prediction is 10 times worse than sex. Okay, but if you go online these days and you follow the Twitter account of our friend Craig Venter, you see that Craig Venter says that they can predict faces of people because he has the Human Longevity Institute and they are super awesome, and somehow they can predict faces, and in fact even showed his own face prediction from his genome. They never published any paper about it. They never tell what algorithm they use. Now, as far as I know, this is the best GWAS of face prediction. And this GWAS basically identified 5 significant variants that explain minute proportion of face variance. Tons of missing heritability. In addition, think about that height. Height is fixed after about 20 years old. It's fixed. There is no age-gene interactions. In faces changes all the time. So your genome that encoded your face while you were babies and while you were 10 years old, 20, 30, it keeps changing. But yet, Venter says that they can predict faces, and there is a lot of hype in the media. Now, I want to claim that if we can only predict minute fraction of the variance of face, we should be over here. You should not see a lot of information. Any face prediction you do could match many individuals. So this is someone that, so here is a person, Jeffrey Reid. After Venter tweeted his face prediction, he thought that this person actually matched Ted Cruz. He used to run against Donald Trump, if you're not from the US, but now he's not running anymore. So this face prediction actually matches. I think that this face actually matches. What's the guy from Wedding Crashers? I think really, I don't know, I see this thing and it keeps annoying me because it seems that something doesn't work with the math over here. We need to have very good predictions. We need to know a lot about the architecture, the genetic architecture of faces to be able to do face prediction that is pretty good. So I think each time that you do something like that, it's very misleading because actually it can fit other individuals, but your brain just looks like, they look similar. But actually they don't. Look like here the nostril, that the nostril is going up over here and it's a bit more pointy, but this one is rounded and the nostril goes down. So I'm not convinced that you can do phenotypic predictions of faces right now. Okay. So we talked about different types of attacks. We talked about surname inference. We talked how sometimes we have these side channel attacks. And then we talked about the limitations of phenotypic predictions. And the question is how we should move forward with these types of genetic privacy. There is a great review in Science about a year and a half ago that talks about human behavior and privacy. And kind of like the bottom line of this review is that privacy is highly malleable. It's context dependent. We ourselves don't know exactly what we want for genetic privacy. If I go and ask individuals over here, let's do that. How many of you care about privacy? Raise your hand. Wow. How many of you use Gmail and let Google read your emails? Okay. How many of you have a Facebook account and let Facebook read your stuff? Good, yeah. So if you raised your hand to the first question, also to the second and third one, there is some dissonance over here. And it's known, it's also, I think I care about my privacy, but I share a lot of things with Google that can be quite intimate. And kind of like I think the fallout of privacy, what we think about privacy, is what happened with Ashley Madison. Ashley Madison was a dating website for married men and women. Does anyone know about it? Does people don't know about Ashley Madison? And okay, people nod their head. I see some participants in there. So Ashley Madison was a website where people it was a website people documented their sexual preferences, they documented who they want to meet. It's a dating website for cheaters. Entire Ashley Madison, all their logs, all their information was leaked about a year ago. 36 million records. And I thought this is really in terms of privacy, that's going to be the end of the world. So, and this, I analyzed actually the names of organs that people mention in Ashley Madison based on their profile, if they are females or males, and you can see what they chat about. And I also wrote this blog about kind of giving tips to preserve the privacy of Ashley Madison users, because really I was convinced that this is the end of the world, and not just me, also New York Times said they thought there was some prediction that Ashley Madison will create a recession because many people are going to get divorced. If they're getting divorced, there will be kind of like a surplus of large apartments, but actually small apartments will be in high demand, so we should maybe buy small apartments to make some money out of it. There was a prediction that the world is going to collapse last August. But now, a year later, 36 million records leaked. Yeah, we heard some horrible, tiny bit horrible stories, but it largely went unnoticed. I don't think there is any spike of divorce rates right now in the US. In Israel, I checked all the email addresses from Israel. It's 200,000 people. We have only 2 million households in Israel, 10%. Yet, it seems, the world is not collapsing, at least not by Ashley Madison. Okay, so kind of my point is that maybe when we have this knee-jerk reaction about privacy, that we think that we need genetic privacy and there is no way that we can move forward because it's too risky that we cannot protect privacy. But I would say that just by looking at Ashley Madison, something that is very intimate and leaked, but still didn't create a collapse, maybe what we want to do is to establish trust relationships with our participants. We should tell them that we cannot protect their privacy, but let's have these long-term relationships. We'll try to protect against harm. We'll be transparent with them, what we do with their data. We'll tell them how their data can benefit medical studies, like this poor girl that I showed at the beginning of my talk. And this way, by working with them and suggesting that nothing in life is risk-free, then we can move forward and create and really share genomic data sets. So to show how it works, we put a website called DNA.Land. I'm not going to talk about this website today because I'm having another talk tomorrow that I will explain how this website works. But we're able to put this website to crowdsource genomic information from individuals, and we obtain now, it's actually more than 22,000 genomes that people around the world uploaded to our website, despite we told them that there is no guarantee for privacy. And we launched this website in October. So basically every day we get on average 100 people that trust us with this website and give us their data. So this is a way basically, and if you want to hear how we establish trust, I invite you to come tomorrow. So the omics session, I'm presenting it 2:15. And this is the acknowledgement slide with my funding, and thank you very much. Questions? Questions about Ashley Madison? Microphone there, yeah.
Hi, my name's Ainsley Newson. I'm from the University of Sydney in Australia and I work in bioethics. Thank you so much for your presentation. Apologies for my husky voice. I was wondering, just regarding the end point of your presentation where you started talking about really, I think, a wholesale rethink about privacy, and I'm wondering about the impact of that publication so far, in the sense that, firstly, I guess, do you think this is something that is exceptional to genomic information? Or do you think we actually require a wholesale rethink about privacy per se in a research context?
Yeah, I think it's a great question. There is some notion in the public that DNA is exceptional in some way, that, oh my God, my DNA can reveal more. People tell me you can learn about ancestry from DNA, but you can learn about ancestry just from looking at Facebook profiles of people in general. So I think kind of like demystifying a bit the way that genomic information works, and telling people that there is a lot that we don't know, but probably you already put so much data out there anyhow, and there are concrete and tangible benefits for sharing data. You can help other families, whereas when you post your Facebook data, you help advertisers on Facebook. I think using this way and really kind of like understand their fears and their concerns and working with them, educating them, and help them reach the right conclusion, this is the way to move forward. There was another question over here.
Tara, you had a question.
Yeah, sorry. Where?
Just quickly.
Yeah.
Please.
Well, here. Yeah.
It sounds like it's safer for women, at least.
Yeah, women.
We're harder to find.
Yeah, you are harder.
In many ways, possibly.
Yeah. Yeah, our method works only for the Y chromosome, but if you saw the pedigrees, since in genetics usually we work with families, once we identify the male, then we can move, and we can also identify the female because they share, of course, the same surname. But this technique only works if you have a Y chromosome.